Hello everyone, my name is Monica Mesecar and I'm a neuroscience researcher here at the University of Michigan. I'm excited to tell all of you about my research today, but before we do that, I thought it would be best to take you on a tour through my research lab so you can see where the magic happens before I start getting into it. Alright everyone, so to begin our tour, as we come down the hall, you'll see that our lab is housed in the Pharmacology department. Pharmacology is the study of how drugs interact with the body. Turning the corner, you'll now see the first shared equipment space. This room contains a giant sink, industrial dishwasher, and a super hot oven. All of this machinery helps us sterilize our equipment between experiments. Coming into the next room, you'll see another shared space that we call the core. Inside the core, there are several industrial fridges and freezers that allow us to safely store temperature-sensitive chemicals and solutions, as well as DNA in cells. There are also incubators for things that need to be kept warm. Lastly, up here at the front, you'll see shakers containing large beakers full of liquid. The shakers help us keep things moving and are temperature controlled. These beakers likely contain bacteria and growth media. Bacteria is commonly used in our experiments because they are easy to grow and manipulate to make the DNA and proteins that we want to study. Turning to the left, we now enter our lab area. As you come in, you'll see more sinks, chemical and equipment storage, and benches for conducting experiments. Here's my bench. It's important to keep your bench as neat as possible and keep your lab coat nearby for safety. Now that you've seen the lab, let me introduce you to our lab members. First up is Dr. Paul Jenkins. Paul is the lab's principal investigator, or PI for short. A PI determines the big picture of the lab, deciding on the general direction of the research, and mentor students within the lab on their specific projects. Next is our lab manager, Leah. Leah has one of the most important jobs in the lab because she is responsible for knowing all of the experimental techniques that we use, as well as keeping up the general maintenance of the lab, such as ordering equipment and materials. Over here is Renee. Renee is a scientist that has been specially trained in this field of electrophysiology, so if any of our experiments use that, we come to him and he helps us. Now down here are Kendall and Karina. They are two of our PhD students, which means they are working on their own independent research projects within the lab. Over the next few years, their goal will be to publish scientific research articles and to write a dissertation detailing their experiments and their results. Last but not least, down here in the wheelchair is yours truly. I am what's known as a post-baccalaureate researcher, which is just a fancy word for in between college and graduate school. I worked in this lab for about a year on my own project, which I will explain shortly. My goal was to gain more research experience and skills before going on to more school. I hope to pursue an MD PhD, which will train me to become both a medical doctor and a PI like Paul. I also want to acknowledge the undergraduate students that help us with our research projects. They aren't here in this picture, but their efforts are critical to our success as a lab. Now that you've seen our space and know a little bit about who we are, let me share with you what we do. As I mentioned earlier, we are a neuroscience research lab. Neuroscience is the study of our brains and how it interacts with our bodies as well as the surrounding environment. Our brains look like this, and different sections of the brain are responsible for different functions. The front of our brain is called the frontal lobe, and it deals with all of our thinking, planning, and problem-solving abilities. The back front part of our brain is called the parietal lobe, which controls all of our movement and spatial planning. Then the sides of our brain are called the temporal lobes, and they help us with our hearing. And then the back of the brain is called the occipital lobe, and that helps us with our vision. This bottom part of the brain is called the cerebellum, and it deals with our movement, planning, and coordination. And then this stick structure down here is our brainstem, which deals with all the functions that we have no control over, such as our breathing. Our brains are made up of special cells called neurons, which look like this. Neurons send signals back and forth between our brains and our bodies so that we can accomplish all of the functions I mentioned earlier. Neurons can facilitate this communication thanks to their unique structure. Let's take a closer look. This part of the neuron that looks like hair are called the dendrites. Dendrites receive signals from the brain or other nearby neurons. The neuron's head is called the soma, and that's where the neuron makes sense of the signal it just received. Next, the signal travels down the neuron's body, called the axon, and ends up at its tail, the terminal, where the signal gets sent out to other parts of the body. But the signal can't travel all the way there without any help. How does it make it all the way across? Located along the neuron's axon are these special proteins called sodium channels. Sodium is responsible for helping carry the signal down the neuron, and sodium channels are in charge of deciding whether the sodium molecules are let in or out. So, sodium channels are an important part of the neuron structure that helps it to do its job. Without sodium channels, the neuron would have a very hard time helping our brains communicate with our bodies. In order to make sure sodium channels are where they're supposed to be, helper proteins called ankrins anchor the sodium channels in place. Our lab is interested in how ankrins and sodium channels work together inside of neurons to promote proper communication. Both ankrins and sodium channels are made according to our genes, the unique instructions that each person has for making everything in our bodies. While many people have similar genetic instructions for making ankrins and sodium channels, some people have instructions that are different than we might expect, called mutations. Most mutations don't have any effect on an ankrin or sodium channel's ability to do their job. But other mutations impact a neuron's structure and function, meaning that these neurons will look and act differently than most people's. Because of this, mutations in ankrins and sodium channels have been connected to conditions such as autism and epilepsy. My research project investigates whether some of these mutations change how ankrins and sodium channels bind together, and what such changes would mean for a neuron's structure and function. We look at binding changes by introducing these mutations into bacteria, isolating the mutated ankrin or sodium channel, binding it to its unaffected partner, and analyzing the interaction using a special machine. This machine will tell us whether the proteins are binding tighter or looser than usual, which may give us clues about how these mutations may impact a neuron's ability to communicate. To examine structural changes caused by mutations, we isolate neurons from mice like these, and we look at them under a special microscope, as shown here. We can then compare the mutated neurons to unaffected neurons to see if they look any different. We hope that by understanding exactly how these mutations may change someone's neurons, we can also better understand and help people with these conditions. Yet, there are differing opinions on how we can best help these people. Many scientists believe that the best approach is to develop drug treatments or gene therapies to reverse or stop the effects of these mutations, effectively curing people. But some of the actual people with these conditions, particularly those with autism, may think that these differences make them unique, so they do not wish for a cure. Given these two conflicting opinions, it raises important questions for how we should direct our future research efforts. Do we push our efforts toward the miracle drug or cure, hoping to eradicate these conditions altogether, or do we prioritize addressing the concerns raised by those living with said conditions? As a neuroscientist who also happens to have a neurological disability, I tend to think that the scientific community has an ethical obligation to center the needs, wishes, and desires of the populations impacted by their research. Otherwise, can we really say that our research efforts are truly helping others? I tend to be motivated by the potential of achieving a greater understanding for understanding's sake, in hopes of being able to better empathize with and support those around me who may be impacted by these conditions, as opposed to simply chasing a cure. These topics have more nuance than can be adequately addressed by this video, so I encourage you to think about these questions for yourself and form your own opinions. For any future scientists out there, your perspectives on these questions may serve as a guiding principle for the type of research you choose to do in the future. I hope you enjoyed this glimpse into life as a neuroscientist and that you learned something from my discussion about my research. Thank you so much for clicking on this video, and I hope you always stay curious.